Well, good day, everybody. This is Chris, and I'm back to talk more about GABA and glutamate. And eventually, we'll talk about barbiturates and benzodiazepines. So we're just laying all the foundational work. And I'll be doing some videos where I bring out the molecular models again, like I did when talking about the opioid molecules. So we'll get some some hands-on, and I'll try to do some chemical reactions as well. But that'll be in subsequent videos. Okay, so when we talk about creating a glutamate and GABA, GABA is actually a byproduct of glutamate metabolism. So what I'm going to do is I'm going to talk about how we produce glutamate and then how glutamate is metabolized into GABA. And what actually happens is this cycle develops, and this cycle of creating glutamate, creating GABA, and then having the GABA break down into other intermediate molecules is collectively known as the Gaba Shunt. And so we're going to talk about some of the basic components of this GABA shunt here. So GABA is produced endogenously, and it comes from endogenously. That just means it's produced within the neurons themselves. It is stored in vesicles, like most other neurotransmitters are stored in presynaptic vesicles. The vesicles are then leave they move toward the end of the axon via exocytosis, and this process is mediated by the presence of calcium. So the presynaptic neuron, and you know what the heck, I'll just draw this here. So the presynaptic neuron and then the postsynaptic dendrite here, and then I have my synapse right here. So I have my neurotransmitter vesicles stored within the axon here, and what happens is the presynaptic neuron depolarizes. All right, so that's sodium going in and potassium rushing out. That's at wave of depolarization. Once it gets closer to the end here, I have voltage gated calcium channels right here. And the calcium channels then allow calcium ions to rush in to the neuron. And once calcium ions rush in, that actually causes the exocytosis, the movement of these little vesicles here, these little vesicles full of the neurotransmitters, and the vesicles move out and they meld with the cell membrane, and then they open up and they release the neurotransmitters out into the synapse, and then the neurotransmitters act as ligands for the GABA receptors. Okay, so that's pretty typical for most neurotransmission. That's a pretty typical thing that we see. So the GABA itself is manufactured inside of the presynaptic neuron, and I want to go ahead and talk about how that happens. So let's just go ahead, and I want to erase all this stuff here. All right, since there's nothing real special there. All right, so what do we get? Well, what I want to do is I want to talk about the Kreb cycle real quick. So I'm going to just start here, and this is a circular process. So it's not like there's a start or an end point. It's rather circular, as you'll see. But I'm just going to start here at the tricarboxylic acid, or the Kreb cycle, or some people will call it the citric acid cycle. All right. Whatever you want to call it. There you have it. Okay, and there is a molecule that's produced in that cycle known as alpha ketoglutarate, and this is actually something that you probably, if you've taken undergraduate anatomy and physiology or cell or organismal biology courses, you've probably heard of this before, because this is This is a substance that we find very common. It's very common to the Krebs cycle, and that's alpha ketoglutarate. So I'll just go ahead and draw that here, or rather put that there. Alpha keto. Let's see if I can spell this right. Keto, Glutarate, I believe. Alpha ketoglutarate. We know this comes from the Krebs cycle. All right, so I have alpha ketoglutarate, and as you find, as I've talked about in other videos, boy, oh boy, is the Krebs cycle so important to so many processes inside of the cell. It's just amazing. It's kind of mind-blowing. You learn about it's noteworthy enough that the Krebs cycle produces the protons and the electrons that we need to run oxidative phosphorylation, or the electron transport chain. That is amazing enough. But all of these other intermediate molecules and cofactors that are used in the Krebs cycle are so critically important throughout the entire cell. It's just amazing how relevant understanding the cellular energy production is, because all of these intermediate molecules and cofactors have multiple jobs. They wear multiple hats. So this is something that comes out of the Krebs cycle. And so what happens is this here is metabolized into glutamate, specifically the L isomer, the L enantiomer of glutamate, L-glutamate. All right, and it is done. There is an enzyme that helps. Well, obviously there's a lot more than just a enzyme or a cofactor, but the major thing that happens there is this reaction is catalyzed by a couple of noteworthy, I guess a couple of noteworthy molecules that we'll talk about. Pyridoxal phosphate being one. So I will just write that there. Pyridoxal phosphate, Pyridoxal, I believe. Pyridoxal phosphate. All right. And we can just abbreviate that as PLP. How's that? Yeah. So pyridoxal phosphate, PLP. L-glutamate is just think of that as that's the glutamate, the amino acid that acts as a neurotransmitter that we all know. Okay, so yeah, so here we go. We have glutamate being produced through this pathway. This is also catalyzed. I won't even go there quite yet. Let's just stay on the alpha ketoglutarate to L-glutamate. Okay, so pyridoxal phosphate, or PLP, is heavily involved in this process. And another thing that's real noteworthy here is that we need vitamin B6, okay, as the cofactor, and vitamin B6 is, you guessed it, pyridoxal phosphate. How about that? All right. So vitamin B6 is a very important, or pyridoxal phosphate is a very important cofactor, all right, in this reaction. Okay, now that I have glutamate, so now that I've taken this common molecule that comes out of the Krebs cycle, turned it into glutamate, glutamate can be metabolized or turned into Gamma-Aminobutyric acid, GABA. All right, and this is the enzyme that helps catabolize this process. The enzyme, all right, the enzyme involved is an enzyme known as glutamate acid decarboxylase. So what I will do here is I'll write that. Glutamic acid, which is just glutamic acid decarboxylase, okay, of glutamic acid decarboxylase, or GAD. We can call that GAD. And this is also a cofactor, PLP. Okay, vitamin B6 is also needed in this process as yet the other cofactor. All right. So what happens here is, and I'm going to draw this here. I'm going to draw the glutamate molecule, and then I have some mock-ups that I'll show you guys here in subsequent videos. So I have H2N here. All right. I'm just going to kind of shorthand this. Each one of these is a carbon, and you can assume they're oxygens attached to where they need to be, but I'm not going to, or hydrogens, excuse me, I'm not going to put all of the hydrogens in there because they're just not really relevant. All right, here we go. All right, so I've got, this is glutamate here, this is L-glutamate, and what I've got here is I want to focus right here on this carboxyl group, okay? And if you look at the name of this enzyme, you can assume what's going to happen to this carboxyl group here. It's going to be decarboxylated. I'm going to remove it. That's the beauty of enzymes, is a lot of the enzymes, what they do is actually in their names. So what happens is when I decarboxylate glutamate, that leaves me with this, H2N, and then I carbon chain capped off with a carboxyl group here. Okay, so I have just cleaved the carboxyl group off, and I have gone from glutamate here to GABA here. Just the difference of that carboxyl group has turned, this is the most common excitatory neurotransmitter in the brain, and now I have the most common inhibitory neurotransmitter in the brain. It's amazing what the replacement or the addition or removal of a functional group can do to the properties of a molecule. That's why learning functional groups is so important later on in biochemistry. Okay, so now I have my GABA. Well, GABA, okay, GABA can be metabolized to succinic semialdehyde, and so I'll write that there, SUCCINIC, succinic ALDEHYDE, and this is commonly called SSA. All right, and what happens is the enzyme that catalyzes this, so I'll put enzyme that catalyzes this reaction is GABA transamase. So let me write that out. TRANSAMINASE, transaminase. Okay, so that's the enzyme that catalyzes that reaction from GABA to succinic semialdehyde, and then succinic semialdehyde is metabolized into see here, succinic acid, I believe. Yes, it is succinic acid. Succinic acid here. All right, and you should be familiar with succinic acid because succinic acid is a molecule in the Kreb cycle, and you can feed that back into the Kreb cycle, and then out pops alpha ketoglutarate, which can be turned back into glutamate, which can be turned into GABA, right? Or, you know, it can be used. Maybe the glutamate's used. Maybe I don't need to make GABA. So it's a really cool thing. And the enzyme, okay, so here was enzyme 1, glutamic acid decarboxylase, or GAD, which catalyzes glutamate to GABA. And then I have GABA transamase, which catalyzes GABA to succinic semialdehyde. And then the other enzyme, so I'll underline that here since these are the other enzyme in this, the major enzyme in this shunt is a succinic semialdehyde. I'm just going to call this SSA, succinic semialdehyde dehydrogenase. All right, so you know what we're doing is we're pulling a water molecule out of there. All right, and this is collectively known as the GABA shunt. This is how we make glutamate, L-L-glutamate, and GABA. And this is how these can be metabolized into other molecules. And you can see that it's circular, and it really crucial to it is the Kreb cycle here. It's kind of like the cornerstone, if you will. Okay, so now that we know how GABA is stored, what causes it to release, and how it is produced, both GABA and glutamate, we should probably talk a little bit about how it is how it's terminated, specifically. So termination. And there are one of two things that can happen, okay? There are one of two things that can happen. GABA can be transported, all right? It can be transported into a glial cell, which is a support cell to the neuron. The specific glial cells that that the GABA gets transmitted into are the astrocytes. So I'll just go ahead and throw that up there. Are the astrocytes, okay? And we'll talk about what happens there versus the other thing that can happen is GABA can also be reuptake. There can be reuptake of the GABA back into the neuron. Okay, you can have reuptake back into the presynaptic neuron. This occurs. There's an actual channel it's a designated channel, and it is actually, just for completion's sake, it's a sodium dependent. It's a sodium dependent transporter. Okay, obviously this is active transport, so your cell's using ATP to do that. Okay, so if the GABA is taken back into the astrocyte, if it's taken into the astrocyte, so okay, what happens to it in case one, and you know both are happening at the same time, but what happens to the GABA that goes back into the glial cell? Well, GABA is, as you might guess, it is like we talked about in the shunt earlier, it is metabolized into succinic semialdehyde, and then SSA is further metabolized into succinic acid. We'll call that SA, which then can be fed back into the Kreb cycle. So that's the fate of the GABA that goes into the astrocytes. The fate of the GABA that undergoes reuptake, if there's reuptake, then, well, hey, not a bad deal there. If there's reuptake, then it can get reused. Simple as that. All right, can get reused. So that's how we have a termination of GABA. All right, guys, I think we are now at a point where I can quit doing the whiteboard videos, and I will actually start filming some molecular videos and show you some examples of the molecules. And I will start by discussing the barbiturates. I'll actually do a quick video where I'll talk about the I'll show you models of the neurotransmitter the glutamate and gamma-aminobutyric acid molecules. So I'll do that in the next video. In the video after that, we will talk about barbiturates. I'll show you a few examples. And then we will finish off this series of discussions talking about benzodiazepines. And there are a whole bunch of them, and I'm not gonna be able to do a molecular makeup of every benzodiazepine, just like the opioids. I will choose some of the common ones that we see in clinical practice. The good thing is the benzodiazepines, just like the opioids, do share very similar characteristics, and we'll talk about what the central structure of those are in subsequent videos. Okay, guys, hopefully you're enjoying these. Hopefully these are making sense, and we'll see you on the next video. As always, thanks for hanging in there.